Hello everyone. Today we'll discuss one of the important topic of CSIR NET syllabus, that is Unit 13 methods in biology, and these are the important for your research purpose, and this technique you have to use after the joining in as a PhD program or in research program. So this questions is the I'll discuss all the previous year question of the related to molecular biology and techniques related questions. So first see the first question. So a researcher was repeating a FACS experiment but somehow got confused with the leveling of the tubes, and there are four tubes, one controls with no fluorescent level, one standard, that is H1 with FIT-C level, and one and a standard, that is H1 with FIT-C level, FIT-C level, and one standard 2, that is S2 with PE level, and the last one T, which should be FIT-C positive, and which the given below is the result of the FACS experiment. Now you have to identify from which what should be the correct leveling. Now a researcher is doing a FACS experiment and he somehow got confused with the tube, right? Leveling of the tube. So here is that first is one control. There are four tubes. Suppose these are four tubes, 1 2 3 4. Sometimes the question is said he forgot the leveling of the tube now, and yes, one was written as control, one kept as control, and the standard one, that is H1 standard one, it's a with FIT-C leveling, FIT-C positive, FIT-C positive, and S2 standard 2, that is P positive, P positive, and the last one, that one test T, which should be FIT-C positive, T also FIT-C positive, T also FIT-C positive. Now we know that during FACS experiment, and we got the result in four quadrant. And what is the significance of that fourth quadrant? Suppose these are the that this is a four quadrant, and four quadrant actually represent, the first one represents the positive and negative, that is one positive for either FIT-C positive or PE positive, right? Either one. And second quadrant represents the both are double positive, and third quadrant represents negative and positive. It's a maybe the PE positive, FIT-C negative, right? And when the left one represent both control, that is a negative for the both, right? These are actually you have to know that the these are the quadrant FIT-C, FIT-C versus P. So now you can see that option. And now option, first look at that option A. That's this is the FIT-C, FIT-C, and these are the one negative and one positive, right? That maybe now first one you can see that the control, you know, the control is double negative, right? Control is there is no fluorescence level, right? Either FIT-C or P, both control is both negative, so it's C should be the control, C should be the control one with no fluorescence level. That means the control, this one should be control, D1, D should be control. So D is C here, D C here, right? Now double positive, both positives positive. Now double positive quadrant, there is no view. So see that first one, this may be the FIT-C positive, right? FIT-C positive. So it's FIT-C positive. So S1 is A, right? A or T here, and S2, you can see that S2 is the S2 will be either PE positive. That means the first one, that's this one is a B, that is PE positive, right? And T is the S1. So answer is the option 4 is the correct answer, right? So you have to know that if you are performing FACS experiment and you got the data result, and you have to put the result in a 4 quadrant, and 4 quadrant you have to know that this represent double positive, this and represent control double negative, and this represent positive negative, and this represent negative and positive, right? And suppose you here fit C and a P is given, and in case of, you can study it with a CD14, CD4, any fluorescence level antibody you can use, and from that data you can analyze the result, right? And now you can also so CSIR also never asked the previous question, but they will ask the different types of related question, right? So you should also know that the in case of apoptosis, the quadrant of apoptosis, like in case of if you perform the FACS experiment, that is on the basis of PE, for study of the apoptosis. Apoptosis is a programmed cell death, right? And for during the study of apoptosis, either the cell is apoptotic cell number or necrotic cell number, we use for that study the antibody, that is annexin 5 and annexin 5 and pi, right? Annexin 5 and pi. So first quadrant, actually those are four possibilities are that necrotic cell, necrotic cell, necrotic cell, late apoptotic cells, late apoptotic cell, and early apoptotic cells, early apoptotic cells, and live cells, right? Now in case of if we plot the annexin 5 versus pi, and here the annexin 5 fit C, and propidium iodide annexin 5 fit C, and pi y quadrant, we will plot the pi propidium iodide, then we'll get the double positive quadrant, that is late apoptotic cells will come here, late apoptotic cell will come here, early apoptotic cells, early apoptotic cells, and these are live cells will come here, and in case of necrotic cells will come here, necrotic cells will come here, right? Now in case of if you perform the FACS experiment, then another important property you should know that these are for the apoptotic cell, apoptotic cells, right? And in case of if you perform the whole blood experiment, and in case of whole blood experiment, if you got the sorry result, and from that you will able to analyze the data also, right? In case of if you perform the whole blood experiment, and then you will, in one axis you will get forward scattered, and then y axis you will get side scattered, right? And we know that during FACS experiment, you also know that part, the the complexity, internal granularity or complexity of the cells represents the side scattered. The higher the internal complexity of the cells, it represents side scattered or SSC, right? Side scattered. And the shape of the cell actually represent the forward scattered, right? Forward scattered, forward scattered actually represent the shape of the cell, right? And side scattered actually represent the internal complexity or granularity of the cell. Now if you perform the whole blood experiment, and then the blood cells will come for, first is the from the this portion, they will come lymphocyte first, lymphocyte, and then monocytes, and the large portions will last come here, the granulocytes. So granulocytes, here we will get granulocytes, Like neutrophils, most of the basically in our blood cells, most of the number is highest is neutrophil. So neutrophil will come here, or granulocytes will come here. Granulocytes. These are monocytes, and these are lymphocyte. So first one for lymphocyte, these are for monocytes, and these are for granulocyte, right? So here you can see that the because CSIR may ask the different experimental based questions, so you must be ready for that, because they will not ask the exact the previous year question, but they will ask that in twist, the making the twist in the question, so that you will have to, the new question will come up, and so you also should know that the late apoptotic, early apoptotic cells, if you use the for apoptosis study, if you want to study the whole blood cells, then we'll give the facts graph in facts data like that, and for apoptosis cell we'll get right here, right? Now we'll see the next question. Now, a cancer progress several genome rearrangement, including translocation, deletion, and duplication, etc., occurs. If these rearrangements are to be identified, which one of the following technique would be most suitable? Now, first of all, in case of cancer cell here, the question is actually asking for that there is a including there is a different genomic rearrangement there, that the chromosomal change, translocation, deletion, duplication occurs, right? Now you have to as a researcher, you have to identify that which chromosome actually the deletion occurs, or then you have to use this different suitable technique. Now here option A is given, RAPD. RAPD stands for random amplified fragment length polymorphism. Fragment length polymorphism. Now, DNA, in case of microarray, we use the microarray, actually, it's a microarray for gene expression study, gene expression analysis, gene expression. Actually, we use the microarray for thousands of genes simultaneously. In one experiment, we'll get the how thousands of genes expression level, right? We use microarray. Now, in case of flow cytometry we use for cellular analysis, not for the chromosomal rearrangement or chromosomal changes. We use the multicolor FIS. What is multicolor FIS? FIS stands for fluorescent in situ hybridization. Fluorescence in situ fluorescence in situ hybridization. Fluorescent in situ hybridization, it's a FIS, and we can also use the for genome wide association, that is related to another term is GISH, that is genome in situ hybridization, right? Now what actually we do during FIS experiment, and first is dividing cells. So answer here is the multicolor FIS. Multicolor FIS is the correct answer, right? Now, in case of dividing cells, what's actually we do during FIS experiment? Suppose there is a first is the dividing cells with metaphase chromosome, because the we study the chromosome metaphase is easily visible with metaphase chromosome. And now a sample of dividing cells, is dried in microscope slide. First we dried in on the microscope slide and treated with formamide, so that the chromosome become the denatured, right? And the do not lose their metaphase morphologies. Then we add the denatured chromosome, and then we add the probe, and probe is detected, right? Then we denaturation, because denaturation, and adding probe, add probe, and probe is detected on the film, probe detection, right? From that we can analyze the where the actually chromosomal translocation, deletion, and duplication and is happened, right? So these are the signal from this for that fact phase is the use for chromosomal translocation. So and then